A very pleasant good morning to one and all gathered in this cyberspace. Research greetings from Jamal Mohamed College. Respected resource persons, delegates, and participants, I am happy to begin the proceedings of second day of National Webinar on Molecular Phylogeny, Evolution, Taxonomy and Ecology, organized by PG and Research Department of Botany under DBT Star College Scheme. We are having two eminent scholars today. The first lecture in the morning will be delivered by Professor Latha Rangan, who is already available on screen, and another lecture will be by Dr. S. Susairaj. Now let us enter into the formal proceedings of the second day. I invite Dr. A. Shah Jahan, Associate Professor of Botany, Dean of Research and Consultancy, Jamal Mohamed College, as well as Sir is the coordinator for DBT Star College Scheme of our college. May I request Dr. Shah Jahan sir to welcome the gathering as well as to introduce Professor Latha Rangan to the audience. Over to Dr. A. Shah Jahan.
Thank you, Aslam. Am I audible, Aslam?
Yes, sir. Very much audible, sir. You can go.
Okay, thank you. Pleasant good morning to all. We are in the second day of the national webinar on molecular evolution, taxonomy and ecology, organized by Department of Botany, Jamal Mohamed College, Tiruchirappalli, under DBT Star College Scheme. In this regard, it is my immense pleasure to introduce and welcome our today's resource person, Dr. Latha Rangan, Senior Professor, Department of Bioscience and Bioengineering, IIT of Guwahati. Madam is going to deliver a lecture on molecular biology, molecular phylogeny of ginger BSc. On behalf of our college management, principal, Dean of Science and Head of the Department of Botany, faculty members of Botany and my own, I express our hearty welcome to Dr. Latha Rangan. Welcome, ma'am. Regarding our resource person, Dr. Latha Rangan, she has been working as the Senior Professor in Department of Bioscience and Bioengineering in IIT Guwahati. Her research area of specialization is applied biodiversity with special reference to bioresource of Northeast India. She has guided more than 13 PhD students and equal number of MSc students. She has published over 40 articles as scientific papers, book chapters, conference proceedings, and reviews. Madam is an elected fellow of National Academy of Science, Allahabad, and Biotech Research Society of India. She is the recipient of awards such as Professor Hillal Chakravarti Award of Indian Science Congress, Young Scientist Award of Society for Science, Chemist and Biologist, Dr. J.N. Bara Science Award for her significant contributions in areas of plant science. So this is a very brief biodata of Dr. Latha Rangan ma'am. Now, actually the primary goal of this session is to understand the basic knowledge about plant molecular evolution. So this is one of our objective of this webinar. And so madam is going to explain the important aspects of this molecular evolution with a special reference to GGPRCA. I hope all of them will enjoy this session. Be ready for note your queries. Once again, I welcome Dr. Latha Rangan. Now the session is over to the resource person. Thank you very much. All the best.
Ma'am, madam,
you
you are
can
in mute,
unmute your
ma'am.
mic, ma'am.
Unmute your mic, ma'am.
I'm audible now?
Yes, ma'am, very much.
Yeah, okay. So thank you very much. And at the outset, I would like to thank the organizers for giving me this opportunity to deliver a talk and then also to share our research experience, which me and my group have been carrying out in IIT Guwahati over the last more than a decade. Yeah, so I joined IIT Guwahati in 2004, and it's been 18 years of footprint for me in IIT Guwahati. And so when I joined IIT Guwahati, my whole focus was to work on something which is specific to the region. And that's when we landed, and we thought that it would be a good idea to work on the family Gingiberaceae, because the Northeast region houses a good number of Gingiberaceae plants. So I'm just going to, just a minute. Yeah, so I hope my slides are visible to everyone. Can you please confirm?
Yes, ma'am, visible, ma'am.
Yeah, okay, fine. Right, so as I said, the title of my today's presentation is Molecular Phylogeny of the Gingiberaceae family. And so for the next 1 hour or so, I'm going to talk about the roadmap which we drew more than 10 years back to work on different aspects of the family Gingiberaceae, especially the species which are actually from the northeastern states of India. So with little background that because we are talking about the genetic resources, and since my lab is focusing more on the applied biodiversity, I thought that with little bit of a background on genetic resources, because the genetic resources, according to the International Convention for Biodiversity, it includes the genes of present and potential values for the humans. And so every country has the responsibility to conserve, restore, and sustainably use. The key is sustainably use the plant genetic resources within its jurisdiction. India being one of the mega biodiversity country, following the path of the development, has been sensitive to the needs of conservation and is still rich in a great array of the plant genetic resources. So the genetic homogeneity enhances genetic vulnerability to the pest, diseases, and the climate change. So if the increase of genetic impoverishment is not encouraged, obviously it may have serious consequences, especially when facing a scenario of changing climate, disease, pest attack, so on and so forth. So in terms of plant genetic resources, India definitely ranks 4th in Asia and 10th in the world. Northeast, since my topic is focused on the Northeast and family Gingiberaceae, Northeast is blessed in the sense that it finds a place in the 25 biodiversity spots of the globe, and it houses a large number of plants possessing great value of medicinal and economic importance, of which the Gingiberaceae is of utmost significance. So you can see a beautiful array of the plants belonging to the family Gingiberaceae in the bottom panel of my slide. Let me just put the pointer on. Right. So what was the motivation for us to land in this family? As I have already said, that it possesses a large number of plants of great medicinal and economic value, and therefore it has a great significance. So obviously, we all know there is an increased demand of the herbal products because of its bioefficacy, because of its less side effect. Then the Northeast India definitely houses a rich medicinal heritage of the plants to this particular family. There are very scanty or very less reports compared to the other flowering plants as far as the Gingiberaceae is concerned. And so when we started, we saw that there was not much of the work on its molecular phylogeny, what is its status, what is its classification. So that was very scanty, and whatever informations were available, that those were kind of very misleading. uh, although the plant is of great economic significance, but we also know that most of the time the plant is underground, and the only time when we are able to see the beautiful characteristic features by which we are able to discriminate it or classify it, it is when it is in the full bloom state. So obviously there are issues of lack of seed and poor propagation rate. Many of the species have been very explored to a greater depth, but then there are great number of the species which have not been explored. Many of it remains as traditionally popular spices. And the other aspect, as I said, there are significant gingers of the Northeast India that have been primarily neglected. So considering all these things, it became one of the driving factor for us and a great motivational part of our activity to embark our journey of the research in the family Gingiberaceae. Now, as far as the classification is concerned, the family is divided into 4 tribes: Alpinioideae, Gingiberoideae, Siphonocoeloideae, and Tamigioideae. Gingiberaceae contains previously recognized Hedychiae, Gingiberrae, and Globae. And as was mentioned, India is one of the richest and diverse regions for Gingiberaceae, having 22 genera and 170 species. Northeast region of the India is the zone of the greatest concentration, where we have seen that definitely 19 genera and about 88 species are reported. And so there is definitely an utmost need for studies, not only on the ethno-medicinal importance of the plant, as the environmental scenario of the region is changing drastically due to the myriad number of anthropogenic activities, and of course added to it is the factor of rapid urbanization. So the conservation of the rich biodiversity for ethnobotanical information requires strong measures. And so various ethnobotanical surveys and studies are prerequisite in attaining these goals. So the only classification, although there have been different classification which has been proposed for the family Gingiberaceae, the one which is acceptable was the one proposed by the scientist Kress et al. In 2002. So the classification of the Gingiberaceae into 4 previously recognized tribes, which was adopted from the Larson 1998 and then subsequently put forth by the Kress, is the one which we are following. And the panel which has been boxed are showing the species Curcuma, Hedychiae, and Kempferia, which are the species which we were interested, and we took it forward for our subsequent activities. So this is not to confuse any further with regard to the hierarchy or the classification of the Gingiberaceae. This is just to show that Gingiberaceae belongs to monocot, and it is closely related to the Canaceae, Marantaceae, and there are other associated families in the order Gingiberales, which also includes the Musaceae. So that is what has been shown for the order Gingiberales, and the tribe Gingiberrae has important genera including Curcuma, Kaempferia, and Hedychium. But then these are not the only genera which are limited to this particular tribe, but then there are other genera as well. But our primary focus remains on the 3 primary genera, Curcuma, Kaempferia, and Hedychia. So henceforth, my point of presentation would be more or less focused on these 3 particular genera belonging to the tribe Gingiberrae and all belonging to the order Gingiberales. Now, as far as the characteristic features of the plants are concerned, obviously the Gingiberoideae, the great period of its time, primarily it remains dormant. It grows profusely by means of its rhizomes, which are very fleshy in nature. And the only time when we are able to distinguish them into different genera and different species within a genera is then when it is in a full bloom state. So this particular slide, you can see the one which is given for the Curcuma plant with its beautiful flowering, which is bright yellow in color, and the characterization as far as its ovary is concerned, that it is 3 locular, And the placentation, these are all the basic aspects. It has an axial basal or the free columnar placentation. But then if we have to go for collection of the genera and the subsequent species belonging to these three genera, the Curcutum, Hedychium, and Camphilia, obviously we know that there are problems, and that problems are compounded because of the fact that many of these species grow in a extremely difficult terrains. So the habitat is highly inaccessible. There is a great difficulty, and it's a very cumbersome, laborious process when we go for collection, not only because it is posed by its inaccessible nature of the habitats, but also for the fact that the flowering period of this plant is only during the monsoon season, only when we know that it belongs to the family Gingiberaceae. Otherwise, there could also be a misidentification because it's closely related to the Canaceae and other families in the order Gingiberales. So as to avoid that, it is important that the collection be made when it is in a complete bloom state, and so the growth is only during the monsoon season. It has been primarily neglected for the reasons which I have mentioned, also for the reasons that the flowers are very delicate, very flimsy, they wither off very quickly, and it loses its color, although it comes in a beautiful array of the colors, and it's a visual delight to the eyes, seeing them in complete blooming state, but then it loses its color very quickly. And since most of the time it remains underground part, it bears the short-lived flowers, and so all collection has to be done only during the time when it is in full bloom, that is after the onset of the monsoon period. So having all these limitations into our mind, the problems associated, we thought that the first and core objective of ours should be to have a good collection of the plant species. The plant species which not only have been collected, but then have been characterized in the sense that the systematic status of this species is well known to us. So the first and the foremost objective of our work was to go ahead with the collection of the germplasm, its subsequent maintenance in the greenhouse, further ethnobotanical survey, because we are talking about the species belonging to the family which have the great economical and medical importance, right? So they are medicinal in nature. And then subsequently the classical chromosome count studies, because these are important. This would be the stepping stones to build on our further molecular phylogenetic aspect. So in this first objective, which is the collection of the germplasm, maintenance, and cytological studies, 22 species of the subfamily were collected from their natural habitat across the different states of the northeastern region. And these collected plants were maintained in the departmental greenhouse of IIT Guwahati. A part of the collections, a subset of the collections were also maintained parallelly in the greenhouse of the Guwahati University. So the collection part of it, subsequent identification and maintenance was jointly done in hands and in coordination and with the whole support of the faculty of the Department of Botany of the Guwahati Universities. So the important point is the collections were made during the flowering season, which is actually April to August. So you can see in this particular slide a good number of the plant species for which we have shown the plants in its complete flowering state. And so one can see the beautiful flowers, the pattern, the color, and different aspects of the plant when it is in a completely blooming state. So this slide is just nothing but only a recap of what I mentioned. 22 species collected from the natural habitat from the northeastern states. So from which states they were collected, from which region they were collected. So for curcuma, we had collected 7 species. For Camphilia, we had collected 4 species. For Hedychium, we had collected the 11 species from nearly about 5 different states. A few of the states we couldn't get the collections, the core collections of these 3 genera or the species belonging to these 3 genera. So the next aspect after its collection was the ethnobotanical survey. So the current pharmacological survey, these were actually carried out, conducted by making the field trips when we were collecting these plant species to different parts of the Northeast during the period from March 2008 to August 2009. So the trips were actually made by the research scholars, the students who were working on this particular project, and these trips were made to villages and tribal areas during different seasons, so as to include a wide number of the species of the subfamily. Since the choice of the individual informant to be interviewed was of the great significance to the reliability of the information, all contacts were subsequently established with the local practitioners. So as you can see in this particular slide, the survey involved 13 taxa belonging to 3 genera, Kaempferia, Curcuma, and Hedychium, and various parts of the plants were actually the data were recorded, which are being used as a herbal medicine, although rhizome was the most frequently used plant part. Now, during this survey, another important thing which was quite interesting to us was that we found that the underground plant part for medicinal plants were found to be higher. That was 56%. Then the case where both above ground parts were used together. So the usage of above ground plant parts were less in comparison to the rhizomatous part, and 13 gingiviridae plants were reported that were used in curing 25 different ailments, of which the highest number of the plant species were used for treatment of gastrointestinal disorder, and 5 of them for curing chest and lungs related diseases. Use of the single plant was found to be very high, which was close to around 84%. And so obviously we concluded that a further pharmacological study of this particular and scientifically unexplored properties of Gingiberaceae definitely seems very promising. This work, the ethnomedicinal users, medical users of species of Gingiberaceae from the Northeast India, that was subsequently published in the Journal of Ethnopharmacology in the year 2010. Then once we had finished this, the next aspect was the chromosome number determination. So obviously we went with regard to the conventional rapid squash method that was followed to prepare the metaphase stage of the cell division of the root tips, which were collected from the plants that were maintained in the hydroponic culture. And the preferred timing was in the morning hours, which was 8 a.m. to 11 a.m. So I'm not going into the details of how we went with regard to the chromosome number counting. That has been already specified here in the flow chart. But once we had done the chromosome number determination, we went with regard to the disparity index value, which is the longest chromosome minus the shortest chromosome over that of the longest chromosome plus the shortest chromosome, multiplied that of the 100. And we also went with regard to the coefficient of the variation. So it's given for the Kaempferia, it's given for the Curcuma, and it's given for the Hedychium, the time of the collection. So primarily, preferably it was in the morning hours. So the next few slides are just only the chromosome number count and the karyotypic, subsequent karyotypic information for the species under the each genera. So for instance, this slide gives for that of Kaempferia pulcherrima. The chromosome number of the 9 species of the subfamily Gingiberidae was investigated, as was mentioned earlier, by the rapid squash technique. So the most of the Northeast Gingiberidae were found to be 2n is equal to 21 or 22 or 33 or 34 or 42, 58 and 63. So 3 different chromosome counts were found in for Curcuma, which are actually 21 and 42 and 63, implicating that it could be a triploid, hexaploid and a non-haploid status of the studied Gingiberaceae species with a basic chromosome number of X is equal to 7. For the genera Kaempferia, the counts were found, as I said, 22 and 55, implicating the diploidy and pentaploidy status. So we predict the basic chromosome number of the genus Kaempferia as X is equal to 11, and in the genus Hedychium, the chromosome numbers were found to be 34. The species are natural diploid with the basic chromosome number of X is equal to 17. So as you can see, these are the slides giving the number of the chromosome with the chromosome length distribution, the karyotype information, and the other aspects, the disparity index value, the variation of the coefficient, as I mentioned earlier. Then subsequently, we went for the karyomorphometrical analysis. So the chromosome length was measured, wherein the statistical analysis of disparity index and coefficient of variation were carried out using the Excel program. So it's a very simple process. And since most of the members of the Gingiberaceae possess a very small chromosome, a detailed karyomorphological study by conventional method was not so easy. So the general feature that was noted in the subfamily Gingiberaceae was a wide range of chromosome with very small size in most of the species. However, the chromosome complements in various members differ in minute karyotypical details. So there was a high value of the average chromosome length, which indicates or hints towards its primitive nature. And there was also a high value of disparity index, which signifies the heterogeneous assemblage of the chromosome. Now, I said that the high values of average chromosome length points towards its primitiveness, whereas the lower values for these parameters were definitely found for Hedychium gardnerium, Kaempferia angustifolia, that denote, because a decrease in the chromatin length is one of the factors that is responsible for evolution of the higher plants, and a comparatively higher disparity index that was found in Hedychium chrysolysium or Curcumara jadoria or Kaempferia galanga corresponds to the heterogeneous assemblage of chromosome in this taxa. But comparatively lower values of disparity index was found in the other members, and that shows that there is a general homogeneity found in various species of the subfamily Gingiberaceae. Normally, a low disparity index value corresponds to the homogeneity of chromosome in most of the higher as well as the lower plants. So to summarize, the collection and maintenance of the Gingiberaceae germplasm was done, ethnobotanical cataloging of 13 Gingiberaceae species from 3 genera, establishment for the 9 selected species was done. There was a karyotype formula that was established for the 9 species. There was a high values of average chromosome length that points towards its primitiveness, and high disparity index in 3 of the genera, which shows a heterogeneous assemblage of the chromosome. So we moved to the next aspect. Once we had done this characterization, not only the identification, but then doing the basic cytological studies, we moved to the next part, which was the assessment of genetic relationship at intervarietal, interspecies, and intergeneric level. So molecular phylogenetics definitely have revitalized the taxonomy and systematic of most living groups. And here is where the DNA fingerprinting for evaluating the genetic relatedness among the species of the genera has become an essential tool for characterization and conservation of bioresources. The ease with which the arbitrarily amplified dominant markers, which we call as AAD, such as the rapid random amplified polymorphic DNA, AFLP, which is the amplified fragment length polymorphism, and ISSR, intersimper sequence repeats, could be used to generate vast quantities of data that has led to their rapid application addressing a diverse range of biological questions. So the most significant users of these markers, which we put it as AAD, arbitrarily amplified dominant markers, remains in their genetic mapping, diagnostic fingerprinting, and in the study of genetic resources within and between populations of individual species. And so that gave us a confidence that for the family Gingiberaceae, the collections, core collections which has been made from the northeastern states, this would be a suitable approach, tool, technique with us to do a molecular phylogenetics of the species. So for this, the different genetic diversity parameters were taken into consideration, which have been mentioned here. That includes the observed number of alleles, effective number of alleles, polymorphic bands, heterozygosity within a total population. And so how do we calculate the effective number of alleles or the niche genetic diversity or interpopulation differentiation? Gene flow is spelled out in this particular slide. So I'm not going into the technical details of it, but these are all the important aspects so as to dissect out a genetic diversity studies for important species in a core collection or in a germplasm. So the sub-objective of this was to have the genetic diversity assessment of the cultivated turmeric variety, because we had a very good collections of the curcuma varieties, primarily from the region of the state of the Assam, from where actually I come from. So this slide gives core collections of the material of the curcuma or the turmeric, the common name being the turmeric. So the sampling was performed for 19 local varieties of turmeric that have been collected from 4 different states. And obviously, once the collection was done, it was grown in the greenhouse of the Department of Biosciences and Bioengineering, which was earlier the Department of Biotechnology. And the PCR amplification was carried out using the AAD markers that included the rapid and the ISR markers. So we included 20 rapid and 20 ISR primers to study the genetic diversity assessment. So this summarizes the assessment of genetic diversity parameters, which includes the effective number of alleles, Shannon index, population diversity, so on and so forth, for all the 19 varieties. So successful amplification was obtained for close to around 10 rapid and 8 ISSR primers. And so we got close to around 82% polymorphism with an average polyinformation PIC value of 0.34. So let me not take the details of here, but then we found that the gene flow and the diversity among the populations was in the order that was expected. The fixation index or the F statistics was found to be 0.25, and the principal coordinate analysis or the PCA grouped the populations according to their origin. So, so this again summarizes the analysis of the molecular variance, the rapid analysis among the population, within the population, and subsequently the mantle test, which were carried out for the rapid molecular markers, the dominant AAD palmers, and individually for the rapid markers, individually for the ISSR markers, and then in combination of both the AAD markers, rapid and ISSR markers, what was among the population and within the population. But as I said, at the end of the day, when we did the principal component analysis, the PC analysis, it grouped the populations according to the origin. So the individuals of the Joharat belonging to the state of Assam grouped together, the individuals belonging to the state of Manipur grouped together, the individuals belonging to the state of Arunachal grouped together. And so we had 4 different clusters: cluster 1, cluster 2, cluster 3, and cluster 4. Cluster 4 comprised of the individuals which belong to the state of Arunachal Pradesh. So that gave us the confidence that the analysis that has been carried out is the acceptable one. So to summarize it, polymorphism percentage and genetic diversity was found to be highest in the Joharat variety. Genetic identity, which is the niche genetic identity, was in the range of 0.8 to 0.87, which further strengthens, implicates that the genetic variation among the populations were very less. There is a gene flow and the diversity among populations, and the analysis or the AMOA showed that it was a 25% genetic variation that was observed among all the population. So we then moved into the interspecies genetic relationship, because the first one was only for the varieties of that of the curcuma. So then was the interspecies genetic relationship. So again, the map showing the collection sites of the species of the Hedychium, and so similar analysis was done for the Hedychium. Similar kind of the genetic tests were carried out. So here we had also included, apart from the rapid and ISSR, we had included the another dominant marker, which is the amplified fragment length morphism, because you expect more of the polymorphism when you do, which combines the robustness and the techniques of not only the rapid markers, but then that of the RFLP as well. So the markers were used individually, rapid and ISSR, but then we had also used a combination of the rapid and ISSR. So the combination of the marker resulted in the high correlation values. So similar PC analysis was done. So PC analysis for the rapid ISSR and AFLP data set for interspecies genetic relationship. In case of Hedychium showed 2 discrete clusters. So this is the cluster analysis for that of the Hedychium species. So for cluster 1, they were collected from the high topological areas, and the species which clubbed in the cluster 2, they were collected primarily, they grouped together, which were coming from the similar habitats, that is from the moist shaded lowlands. So this is for again for Camphilia species. Let me not go into the details, but then similar pattern work was followed using a set of the molecular markers. So again, the rapid and ISSR combination grouped the species according to their morphology. Then we come into the intergeneric relationship, where we have all the genera that were collected from the northeastern states. So it includes the Alpinia, it includes the gingiber, it includes the Camphilia, it includes the species of the curcuma, it includes the species of the Hedychium. Intergeneric studies were subsequently performed, and the genetic relationship within the genus Camphilia, gingiber, curcuma, Hedychium, and Alpinia were taken into consideration. So the aim of this study was to understand the genetic relationship patterns of the subfamily Gingiberidae from the Northeast India. And it is done to confirm that whether the rapid and ISSR markers could able to group the members according to their genus or not. So to study this objective, we have taken samples spread across the family Gingiberidae, 4 species from the genus Camphilia, as was shown in the table, 6 species from the genus Curcuma, 6 species from the genus Hedychium, 1 species from the genus Gingiber, and we have also taken 1 outgroup taxa from the subfamily Alpinioidae, that includes the species Alpinia nigra. So 10 rapid and 10 ISSR primers were used, but amplification in all the species could be achieved only for 6 rapid and 5 ISSR primers. But then the ones which gave the successful amplification resulted in more than 90% polymorphic fragments. The highest informative primers for the rapid and ISSR were recorded, and the highest genetic diversity for the subsequent markers were also recorded, which was found more for the ISSR marker than compared to that of the rapid marker. Now, this dendrogram, which is shown over here, the rapid was able to group the species according to its outgroup, because we had included 1 outgroup species, which was Alpinia nigra. So cluster 1 was formed by species from Hedychium. Cluster 2 consists of most of the species from Curcuma and that of Camphilia. Cluster 2 also has been grouped into 2 subgroups, subgroup 1 and subgroup 2, which consist species solely belonging to the genus Curcuma, and subgroup 2, which consisted of the species from the Camphilia. So it was found that the curcuma and hedychium form compact clusters, whereas Camphilia galanga was falling within the genus hedychium. So the results of the cluster analysis was strongly supported by that of our principal component, principal coordinate analysis. So the rapid marker confirmed the intergeneric relationship of the subfamily and formed two groups that I had already mentioned. And our study confirms the placement of gingiber within the tribe Gingiberoideae. Then we move to the other aspect, moving on from the marker studies to that of the another advanced techniques, which is also being now subsequently used for sort of the molecular systematics and phylogenetics, and that is the technique involving the use of the laser system, which is the flow cytometers. So flow cytometric studies on the Gingiberoideae and the species from the other family was subsequently then taken forward. Now, what is this flow cytometer? So flow cytometry, that is the flow of the cells in a liquid system, okay? So flow cytometry has definitely become an important tool for measuring the nuclear DNA content in plant biology with its application in crop and horticulture sciences. It's also useful in the genome size research and other areas of population and evolutionary biology. The DNA is measured and is expressed in terms of the C value. So the DNA C value for the species is nothing, but it is the amount of the nuclear DNA in an unreplicated haploid genome of a gamete. And we refer it as a C value paradox, which has been given way back in 1971 by the Thomas, or the lack of the correlation between the genome size and the number of the genes and the organism complexity. And this is a well-known phenomena. However, the data is lacking in context of molecular phylogeny, especially for the Gingiberaceae, which takes into account of the karyotypic parameters along with the molecular markers and genome size data. So the knowledge of the haploid DNA content, which we call as the C value, is definitely important in studies involving genome organization, species relationship, and germplasm improvement. And this C value is actually represented by megabase pairs or in terms of picogram. So the haploid genomic content is quantified species-wise, phylum-wise, or kingdom-wise in increasing order of evolutionary table. And one can find numerous variation not only in the same phylum, group, order, family, genus, or species, but also in different tissue types of the plants. And this has been reported in 2005 in a very good classical paper by the Zhang et al. So the knowledge of the genome size enables the researchers to progress further in the sequencing aspects, usage of the fingerprinting molecular techniques that I just talked earlier, that is in terms of fingerprinting analyses such as the amplified fragment length polymorphism and also in the cloning strategy. So earlier also plant genome size were estimated, but then it was estimated by a completely different technique which involved a staining process, the Fulgen microdensitometry method. But then, however, the limitations are it's a cumbersome process, a laborious process, it's slow, and it definitely poses a more instrument difficulties, making it less reliable. So the more efficient technique now to estimate the plant genome size is using this laser flow cytometers, the flow cytometry technique. So it overcomes much of the limitations of that of the Fulgen microdensity method because it is fast, it's accurate, analyzes the individual cells or the nuclei, and it is highly reliable and reproducible, making it the choice to use this particular technique to estimate the plant genome size. So there are systematic steps which are involved so as to estimate the genome size of a particular species. So there are different isolation buffers which have been adopted, used in the past, but in our study we found that the nuclear isolation buffer which was proposed by Professor Avtar Krishan in 1975 for mammalian cells. In fact, it was for chick cells that worked effectively for the plant cells as well. So the nuclear isolation buffer contains different components which allows for the release of the cells or the nuclei, so that these nuclei are subsequently stained and allows it to be quantified in the laser flow cytometer. So the components, concentrations of different components are specified. So it contains a chelating buffer, it contains a ribonuclease which would cleave and digest of your RNA, because the interest is only of the DNA, and then it contains a suitable dye. So propidium iodide is a dye which resembles that of the ethidium bromide. It's an intercalating dye. So the process is very simple. It hardly takes 10-15 minutes to process the samples, then acquisition of the sample, running of it in the flow cytometer, and then deriving at the data is a matter of another half an hour. So the young tender leaves are generally chopped in the nuclear isolation buffer, incubated with the RNAs for 30 minutes. It is filtered through the micron nylon mesh so as to remove the unwanted debris, viewed under the fluorescent microscopes so as to ensure that the cells are stained, and then subsequently acquired in the flow cytometer. But then to do this process, because we were working on an extremely hardy system, the family Gingiberaceae, comprising of the species where there are not only, apart from the primary metabolites, there are secondary metabolites also, and presence of these secondary metabolites definitely would hinder and interfere with the results of our work. So what was expected from our side was to first go with regard to the optimization of the flow cytometric protocol, and this is in fact recommended for every species, because every plant species, an ideal optimization has to be carried out so as to get the best results. So optimization was carried out in our case also. When we go for optimization, obviously we need to run some kind of a standards, because you have an unknown sample and you are comparing the unknown sample with that of a known sample for which we already know what is the genome size. So 4 different standards were used: rice, Oryza sativa; tomato, Solanum lycopersicum; maize, Gia maize; and pea, which is the Pisum sativum. And the subsequent genome size of these 4 reference standards are mentioned below. The smallest for that of Oryza sativa, which is the 2C value of 1.01 picogram, and the highest for that of the garden pea, Pisum sativum, which is 2C value of 9.09 picogram. So we calibrated these 4 different standards for our system, that is for the family Gingiberaceae. Now, while doing the optimization, obviously we had to also optimize the kind of the filter mesh we were using. The filter mesh comes either in 30 micron or 40 micron or 100 micron, because you have extracted a crude in the nuclear isolation buffer, incubated with an RNase, and then subsequently filtered it. So we did the optimization of the filtering through this nylon mesh. So the whole objective was to remove the unwanted debris and to get rid of interfering substances, and all that we were interested was the well-stained single cells without any kind of the clump or the aggregate. So in our study, we found that 30 micron to 40 micron worked well for us to get rid of most of the unwanted debris, dead cells, and clumped cells, aggregated cells, which are not required and which would hinder with that of the flow of the cells when passed through the laser system. So I'll skip this part. And then subsequently, the second optimization, the optimization not only with regard to the standards, optimization not only with regard to the filters that is to be used, but then optimization also with regard to the components which are used in the nuclear isolation buffer, especially with regard to the detergents. So we had used sodium acetate, and also we had used the NP40 at a very concentration ranging from 0.03 to 0.3, which was volume by volume. And we found that increasing the concentration of the NP40 from 0.05 to 0.3 obviously aided in breaking the cell wall of many of the species of the Gingiberaceae family, because the whole purpose of this detergent is to break the cell wall so that the contents from the cell, from the cytoplasm, are released into the nuclear isolation buffer. So the concentration of 0.3% was found to be very effective in aiding and breaking the cell wall of some of the tough species of the Gingiberaceae. So these slides are just nothing but a reaffirmation of the 0.3% of the detergent of NP40 for different species of curcuma, hedychium, and Camphilia. And so we found that uniformly 0.3% worked very well for most of the species of the Gingiberaceae. Then was the aspect of the standardization, because although we have a range of the standards starting from as small as that of the rice or Rhiza sativa and as big as that of the Pisum sativum, which is 9.09, but then there are different standardization techniques used. Something called as external standardization, where the sample for which the genome size has to be estimated is processed separately. The reference standard, whatever reference standard is being used, that is processed separately, and then you simulate the results. That is external standardization. But of late, it has been specified that whenever a genome size is being estimated, instead of external standardization, the process should be an internal standardization process. Internal standardization process is nothing but then where both the test sample, that is for which we are going to estimate the genome size, and the reference plant for which we already know what is the genome size, they should be processed together in the same nuclear isolation buffer and should be run at the same time in the flow cytometer. So, so this slide gives that for external standardization process, where the curcuma jadoria and that of the Gia maize, which was used as a reference standard, they were processed separately and run in a flow cytometer separately. And we see the histogram peak, which is the G0, G1 peak of the test samples, which gives us the value of the C value. And subsequently, the values were calculated. While doing the external standardization process, what is the issues? While using the external standardization process, we found that there is a shift in the mean peak position when we acquire it in the flow cytometer. So if it is shifting, the mean peak position is shifting, then that is an indication that there are presence of inhibitors, and that inhibitors are interfering during the time of acquiring the results, binding of the propidium iodide dye to the nuclei. So that means external standardization process actually gives an indication that there are inhibitors in the nuclear isolation buffer. The cleaning has not been effective, and so it could give a result that would be false positive. So the effect of inhibitors could be well tested when doing the external standardization process. While you do an internal standardization process, you should not see a change in the shift of the mean peak position of the reference standard. And so if you're not seeing the change in the peak position of the reference standard, then it is definitely an indication that inhibitors have been overcome. So the bottom two panels are that for the internal standardization process. The top panels are for the external standardization process using the reference standard as Pisum sativum, and here is for the test plant, which is curcuma, Camphilia angustifolia. So that's why the objective was to go for internal standardization process. So to summarize it, we used the 30 micron nylon mesh, which was optimized for the plant flow cytometry. 0.3% of the detergent was standardized for the estimation of the nuclear DNA content. The optimized buffer was found to be suitable not only for the release of the nuclear DNA content, but then also to get rid of most of the debris and the interfering substances. DNA content was estimated using the external standardization initially for Curcuma jadoria, but then we noticed subsequently for the subspecies that it should be adopted and followed with the internal standardization process so as to overcome the effect of the inhibitors or to confirm the presence of the inhibitors. So then we moved with the subsequent aspect, which is actually the variation of the nuclear DNA content in the different tissue types, because the reports have already been suggested that the nuclear DNA content could also vary in the tissue types. So this one shows for that of Curcuma jadoria. So you have the different whorls or the floral parts of the flower. That includes the bright colored petals, then the staminode, and which is the male part, and then the stigma. So we separated out the different parts of each of these flowers belonging to a particular species, and we then did the flow cytometry analysis so as to see the tissue specific variation, not only in the floral parts but then also in the vegetative tissues of Curcuma jadoria. So you can see over here it is for the leaf tip, it's for the root tip, it's for the rhizome, and we noticed using an internal standardization process having 3 different standards that include the Oryza sativa, Solanum lycopersicum, and that of the Gia maize. We found that there was no significant variation in the nuclear DNA content when we were trying to see if there exists any variation in the vegetative tissues. So is the case in case of the floral parts, and that includes the ovary style stigma. For vegetative tissues and for the reproductive tissues, we found that there was no statistically significant variation as far as the nuclear DNA content was concerned. So let me skip this. So similar pattern was done for the genera of Bosenbergia, Camphilia, costus. And so we concluded that the nuclear DNA content vary based on the type of the tissues that are being used for estimation. The dividing parts, that includes the highly meristematic tissue, represented non-significant variation in the nuclear DNA content. The reproductive parts of the plant represent to some extent a different order of the DNA content, which is expected. Then we move to the intra-species variation in the nuclear DNA content of turmeric variety of the Northeast India, where initially we had done the marker analysis using the rapid markers and ISSR markers. So here also we wanted to see if there exists any kind of a correlation with that of the markers, where we are exploring the opportunity to use the tools of the flow cytometry to see there exists any variation as far as the DNA content is concerned in the varieties of Curcuma collected from the Northeast. So the same set of the information, the state of the origin from where these species were collected, and so the 2C value generation using the set of the standards that included the Oryza sativa and the tomato, the Solanum lycopersicum. So we observed that there was a maximum variation that was seen in the turmeric variety of the Assam. So using primarily the standard Oryza sativa. So there was a fold variation in the nuclear DNA content that was highest in the variety of the Assam, that was followed by the Arunachal Pradesh, and we noticed that there was a positive correlation that was observed between the average nuclear DNA content of each population, inferring that the genetic diversity definitely increases with an increase in the nuclear DNA content. So just to summarize this sub-objective, the lowest fold variation was reported for the variety of the Meghalaya, whereas the highest was that for the Assam. The average fold variation was 2.1, and there exists a positive correlation between the nuclear DNA content and the genetic diversity parameters. Then finally, we came into the aspect of the nuclear DNA content estimation for all the species belonging to all the genera or for the tribe Gingiberoidae. So the nuclear DNA content was estimated for 20 species of the subfamily Gingiberoidae by using 4 internal standards. And just to quickly run through it, the minimum value of the nuclear DNA content was observed for that of Curcuma leucorrhiza, that was around 1.6, whereas the maximum value of the nuclear DNA content was observed, and the fold variation was reported to be 4.74 with a narrow but biogeographical hotspot of India. So the fold variation of 2.8 was reported for the study of the genus Curcuma. The nuclear DNA content, which was estimated for Globa, Bosenbergia, Gingiber officinale, Gingiber moran, or the Costus species and Alpinia nigra, is to get a clear idea of fold variation that is persisting in the family. And so the fold variation in case of Hedychium was 2.14. So we not only included what we had got in our report, but then we also collected if there were any references existing for some of these species that has been included in our study, and we compared it with those as well. The ones which are boxed in the red are the ones which are the new reports, for instance, for Hedychium coronaria or for the Hedychium gardnerium as well. But that also indicates the smallest one and that of the highest one. So the fold variation in Hedychium was 2.14. In Camphilia, it was 2.28. The smallest for that of Camphilia galanga, and the highest was that for of Camphilia pulchera too. So the maximum nuclear DNA was found as far as the genera Curcuma is concerned was in Curcuma jadoria, whereas the minimum was found in Curcuma leucorrhiza. So 1.68 and the maximum is 2.78, giving a fold variation of 1.72. As far as the genus Hedychium is concerned, it is 4.3 maximum and minimum in coronarium, which is 1.96, making the fold variation around 2.14. As far as the Camphilia genus is concerned, the maximum was for the galanga, close to around 8, and the minimum in Camphilia pulchera, which was close to around 3.5, with a fold variation of 2.28. So overall, the fold variation was 4.05. Then we moved to the dynamics of the chromosome number, because we had initially done the chromosome counting, the cytological test, chiromorphological analysis for these 19 to 20 species of the family Gingiberaceae or subfamily Gingiberoidae, comprising of the 3 genera Curcuma, Camphilia, and Hedychium. So, and so we wanted to see if there exists any kind of a dynamics as far as the chromosome number is concerned, which is given as in terms of the N, and the nuclear DNA content, which is given in terms of the C value. So we already know what was the chromosome number, and we had also estimated the nuclear DNA content using the flow cytometer. So as can be seen in this particular table, the chromosome number, which is given in as a 2N, and the nuclear DNA content, which is given in terms of its picogram, the 2C value expressed in terms of the picogram for these 9 species, right? So for this 9 species, or for the subfamily Gingiberoidae, what we noticed was that the subfamily Gingiberoidae actually was forming into 3 groups. Those that showed little variation, those that showed a moderate range of a variation in the genome size, and those that showed wide range of variation in the genome size. So most of the taxa are of the first group, that is the class 1, and may be characterized by having either a small genome size, and that includes Curcuma, Hedychium, Globa, The second group, which we call as the class 2, consisted of those species with the moderate range of genome size that included, that included Gingiber, Camphilia costus, and Hedychium, and the third consisting of Camphilia and Bosenbergia, having the highest genome size. So that is what is given over here, the genome size or the 2C picogram value and the chromosome number, distinguishing it into very clearly 3 distinct groups or the clades. So in corroboration with the genome size data, the DNA content, which is often used as a proxy for ploidy in comparative studies of chromosome variation, this is based on the assumption that the DNA content will vary with the chromosome number at fine phylogenetic scales when chromosome number increases due to whole or the partial genome duplication. Obviously, the aspect of the chromosome number increase is because of the duplication. So I just go back here. And so rescaling the data in this way, we have found that expressing the chromosome number and genome size in common unit, but had no effect on the R square or its significance. So what we say is that if chromosome number changes were due to unbiased deletion or duplication of the chromosome, the slope of this regression would approach 1 with increasing data. So if the chromosome number changes are due to fission or because of the fusion with no change in the DNA content, the regression slope will approach the 0. So this was a kind of a statistical analysis that gave us the confidence. And so we use this positive Spearman correlation coefficient for chromosome number and 2C value that suggest that the genome size and chromosome number data are positively correlated. The regression analysis of the chromosome number and 2C value yielded a regression equation, which is mentioned over here. And the near 0 regression coefficient definitely signifies that the chromosome number changes are due to fission and fusion with no change in the DNA content. So just to summarize this aspect, where we are trying to draw a correlation of the chromosome number with that of the genome size. So we say that those that show little variation, those that show a moderate variation clustered in the group 2, and those that showed a wide range of the variation in the genome side had the highest genome size, including the species belonging to the genera Camphilia and Bosenbergia. So, so just to summarize the every aspect of the study involving the applied biodiversity, primarily involving the molecular phylogeny of the Gingiberaceae, I would just summarize saying that the study demonstrated the ethnopharmacological significance, molecular cytogenetic and phylogenetic aspects of the subfamily Gingiberoideae. The ethnobotanical survey is definitely the first of the kind of categorization use of the plants in the traditional medicinal practices. Chromosome count and their associated karyomolecular information of 9 species provided information about the chromosome evolution. The knowledge of the genetic relationship assessment for the wild germplasm of Hedychium, Camphilia obviously would be helpful and it would aid in the conservation and its sustainable use. Flow cytometry protocol, which was optimized for the Gingiberoideae for the first time in the new isolation buffer, involved the studies where the nuclear DNA content was estimated. The variation in 10 different tissue types of 6 different plants has been established for the first time, although the variation was not that significant that it calls for an extensive study. And the genome size data was evaluated for 10 new species using the flow cytometry. Also, the fold variation of the nuclear DNA content in the Gingiberaceae was established for the first time. Altogether, the dynamics of the nuclear DNA content and chromosome number was established for the subfamily Gingiberoideae. So what could be the future directions of the work coming out from the work that we did? Obviously, there is a lot of scope, opportunity to include more of the plant species belonging to the subfamily, because we had a limited number. We included only a limited number of the species which were identified and systematically characterized. And this would definitely then pave the way for much further detailed ethnobotanical study of the region. Obviously, characterization of the gingiviral germplasm can also be further expanded by including more and other useful marker systems. And then obviously there could be the characterization of repeat elements, because from flow cytometry, we obviously could get the information with regard to its genome size, but then what is it which is accounting for the genome obesity or reduction in the number of the chromosome, and also small size of the genome, that could be looked by exploring more of the repeat element analysis. So functional characterization of those repeat elements, the transposable elements, which accounts for more than 70% of the genomic content, would throw much more highlight into the aspects, how they are evolving or how they are downsizing when the process of the evolution is taking place and the species are progressing or evolving to a more robust kind of the environment. So with this, I would like to wind up my presentation. And again, I would like to thank the organizers for giving me this opportunity to share our work with you all. And I would like to also acknowledge my active research group, my students who have graduated out, and students who are currently working with me. And so because we are exploring the possibility of looking into the functional characterization of the repeat element analysis, taking the case of some of the selected species of the 3 genera. We still have long way to go to come out with the conclusive answers to it. But nevertheless, the work being carried out by the students past, present, is highly acknowledged, especially the most important point, which was to do with regard to the collection of the material, which is definitely a very cumbersome process. And then my collaborators with whom I am working on different aspects of systematic characterization and also on the repeat element analysis. So this work was jointly done with a collaborator from the University of Georgia, Professor Jeff Benedson, who is also an authority in that of the repeat element analysis and marker analysis, and Professor Peter Nick, who is from Karlsruhe Institute of Technology in Germany, with whom we are closely collaborating for the species of Alpinia nigra, belonging to the tribe Alpinioideae, although I have not included that here in my present study, except for using it as an outgroup taxa. The funding agencies, different funding agency, is to be greatly acknowledged. A part of this work was supported by Department of Information Technology, Ministry of Information Technology, and also Department of Biotechnology, and all the support that has been extended by my parent institution, Indian Institute of Technology, my department, and all others who have joined on different aspects in this road and the journey which we have taken to explore some facets, some information on the family Gingiberaceae, especially from the northeastern region. Thank you very much.
Thank you very much for this wonderful presentation, ma'am. Okay. Let us look for some quick questions from audience. Ma'am, I have one question in this topic.
Yes, please.
This gingiberals themselves, most of them have vegetative reproduction only. They do not produce active seeds. what is your opinion about this, ma'am? Like, have you come across any species that produces seeds, and have you included them in your studies?
Right. Yes, some species do produce seeds. Alpinia nigra, which I was talking about. Nigra means black. So the seeds are black in color. So there it propagates through the seeds, although the rate of the germination is very slow. So I think the plant propagates primarily through its vegetative parameters, either the rhizomes or the stolons or the suckers, sort of that. But nevertheless, yes, it do have floral parts, and there are reproduction by means of the seeds as well, although the rate of its growth germination through those seeds are very less. You are muted, sir.
Thank you very much for the nice info, ma'am. I think there are no questions in the chat box. Probably we can wind up, ma'am.
Okay.
Thank you very much for sparing your valuable time with us. On behalf of the organizing committee, on behalf of the Department of Botany, as well as on my own behalf, I express my sincere thanks to you, ma'am.
Thank you very much. And please, you can share my mail ID with the participants in case in future also they would like to reach out to me for something. You can always share my mail ID with them.
Okay, ma'am. Thank you very much.
Thank you. Thank you very much.
Have a great day, ma'am.
Yeah, thank you very much.
Now we are moving on to the next lecture of second day. Balaguru sir, are you there? Yes, sir. Okay. Shall we move ahead? Yes, sir. May I request Dr. B. Balaguru, Assistant Professor, Department of Botany, Jamal Mohamed College, to introduce the next speaker, Dr. S. Susiraj, to the audience. Over to Dr. Balaguru.
Welcome all participants. Good morning to all. I welcome Dr. Susiraj, Assistant Professor, Department of Botany, St. Joseph's College, Tiruchirappalli, for this second session of this national seminar on molecular evolution, taxonomy and ecology. Dr. Susiraj has a teaching experience and research experience has 20 years. He is a recipient of Young Scientist by DST Serb. He published more than 109 articles in national and international reputed journals, and he presented papers more than 34 conferences and seminars. He published 9 articles in book chapters. He discovered 5 new species. That is a very wonderful things for his research career. He received 2 major research projects, 1 minor research projects from DST, UGC and Tamil Nadu Forest Department. He guided 5 PhD students. Now presently 4 students undergone PhD under his guidance. He submitted 5 gene sequences to the NCBI. He is a life member of Indian Angiosperm Taxonomy. With this short introduction, I invite Dr. Susiraj, Assistant Professor, Department of Botany, St. Joseph's College, to deliver lecture on molecular tools in Angiosperm Taxonomy. Over to Dr. Susiraj.
Good morning to all.
Dr. Susiraj, please make it to present view. PowerPoint slideshow view.
Yeah. I have uploaded. Is it visible?
Yes,
Dr. Balaguru, is it visible?
it's visible.
Okay.
To make it into PowerPoint slideshow view. Below the PowerPoint, there's PowerPoint slideshow view. Below that,
PowerPoint?
slideshow. It is in PowerPoint.
I have shared it. PowerPoint.
Professor, again share your screen, sir.
லேப்ல உட்காந்து Now it's okay, sir.
Is it okay, Balaguru?
Yes, sir. You can go ahead.
Is the slides are changing?
Yes.
Okay. Thank you. Thank you. I thought it is at 11:45. Okay. So at the outset, I would like to express my gratitude to the management of Jamal College, Tiruchirappalli, and my colleagues at the Department of Botany, Jamal College, Tiruchirappalli, for the opportunity extended to me to be with you and discuss on phylogenetic systematics of angiosperms. I know the faculty of botany at Jamal College. They are very cordial, genuine, and highly professional. And in a special way, I thank all of them. So with this short acknowledgement, I would like to get into the topic. The title of the topic is Application of Molecular Data in Angiosperm Systematics. So the plant taxonomy or plant systematics comprises or mainly interested in naming and classifying the plants. So of these two, now today at present, we will focus on the systematics of systems of classification of angiosperms. Angiosperms, the flowering plants, are pantrophic in distribution, comprises of nearly 190,000 species. And these angiosperms are highly diversified in terms of evolution. It has evolved in Cretaceous period of Mesozoic era around 125 million years ago and flourished in Tertiary period of Cenozoic era. The classification in the system of classification is a system of arrangement or grouping organism where hierarchy is followed from species to genera and to different ranks based on certain principles. These underlying principles used for classification were modified greatly over period of time as the science and technology has evolved. So we could see this slide shows from character of a macroscopic importance. Now we people, the taxonomists, use the characters at the molecular data. So the system of classifications were from artificial system to natural system and the phylogenetic system. It has evolved, and this evolution is brought about by availability of characters of macroscopic to the molecular data. So when the macroscopic data alone were being used, we had artificial system of classification. And as technology has advanced, we have a microscope being invented in the 16th century, and this has provided with a whole lot of numerous data of organisms from anatomy, embryology, karyology, palynology, etc. That is provided as a numerous character which led to the natural system of classification. Furthermore, in the development of the spectroscopic studies like GCMS or NMR on the secondary metabolites led to the system of classification known as the chemotaxonomy. And in the advancement of computers with the morphology and other features that was used in the natural system of classification, there derived another system of classification called numerical taxonomy. At the advent of PCR sequencing technology and bioinformatics, etc., so we have been provided with gigabytes of data, so much of data from morphology, from macroscopic studies, and from palynology. All those data were available at our disposal. So that led to the scientists to develop phylogenetic systems. So as the technology has evolved, the taxonomists have made use of all those technology to aid the system of classification, and it has become more and more complex. So basically, there are three kinds of system of classification recognized: the artificial system of classification, a natural system of classification, and the phylogenetic system of classification. So Linnaeus, in his book Species Plantarum, classified the flowering plants, the angiosperm, mainly based on the male flower and the female flower pods. And naturally, this has been recognized as an artificial system of classification. And a group of scientists like D. Jesse, D. Cantole, Bentham, and Hooker have developed another system called a natural system of classification, where morphological data and all other data have been incorporated. Inger and Brander and followed by a group of scientists like Beste, Hutchinson, Cranquist, Tankagen, they have proposed a system of classification called a phylogenetic system of classification due to influenced by the Darwin's theory of evolution in 1859. So this system was more of a phylogenetic system, and this phylogenetic system was bestowed upon the evolutionary principles. So they had certain concepts, they call it as a principles. Like Hutchinson's, he had 22 principles. Some of them, like trees are considered to be primitive than the climbers. Similarly, epiphytes and parasites are considered to be advanced than their host. Spiral arrangement of flower pods are happen to be primitive than the cyclic arrangements. Unisexual flowers are advanced than the bisexual flowers. Solitary flowers are primitive than the inflorescence. Similarly, actinomorphic condition arrangement of the flowers are considered to be primitive than the zygomorphic condition. So likewise, they have used a set of principles to develop a system of classification called phylogenetic system of classification. At present, we have the angiosperm phylogeny group system of classification. It was introduced in the year 1998. And 3 revisions have been published, followed by in 2003 the APG system 2, and 2009 APG system 3, and 2016 the APG system 4. With the efforts of 2 international group of systematics botanists from Royal Swedish Academy of Sciences and Missouri Botanical Garden, which is a collaborative work of more than 53 taxonomists. The main intention of a angiosperm system of classification or phylogeny group system of classification is to provide a widely accepted and more stable point of reference for the angiosperm classification because the angiosperms are highly diversified and numerous in number. So the APG system, here is the outline, condensed outline of APG system 4, where it is divided into a basal angiosperm, beginning with 3 orders. It is initiate with Amporellus and end with the eudicots solanalis. So the primitive, primitive angiosperm are grouped into basal angiosperms that comprises of 3 orders, followed by magnolids that comprises of 4 orders, and monocots are 11 orders, beginning with Acorellus and ending with Gingiberellus, and eudicots with 43 orders. So the order Chloranthalus is considered to be independent lineage. So it has been placed in between magnolids and monocots. And the order Ceratophyllus is considered to be sister of eudicots. So it is just placed before the eudicots. So the core eudicots, core eudicots as a core eudicots of 39 orders, which is being divided into 2 superclades, super rosids and super astids. Majority of the orders in the super rosids, there are 18 orders, of which have been grouped into a clade rosids comprising of 17 orders, and the rosids are being divided into 2 orders, fabids and malvids. Fabids with 8 orders and malvids with another 8 orders. The superclade, supraastids, comprises of 20 orders, and again it is divided into 2 clades, campanulids and lamids. Campanulids with 7 orders and lamids with 8 orders. So this is a short form of the elaborate system of APG4 system of classification. So I like to present you with some salient features of this APG system of classification. And the APG system was based on the sound phylogenetic principle being constructed using the idea of a monophyly. So all the group or all the taxon, based on their evolution, if they have a common ancestor, been grouped into a family. So there is a relationship within the taxa of angiosperm, and that relationship is called ancestor and descendants. So all the descendants have been grouped into one ancestor, and all the ancestors again grouped into a basal ancestor. So this system of classification was based on MATK and RBCL region of genetic material, which is the maternally inherited, because it is a plastic marker. MATK and RBCL markers are plastic markers found in the chloroplast. So they are inherited maternally, mother based. So that the nuclear based genes or markers have been omitted because they get into a sexual recombination during sexual reproduction. So that is being avoided. So in the APG system, the family is considered to be the central point, and hence more emphasis is being given to monophyletic evolution of a family. As a result, APG4 comprises of 64 orders and 416 families. So we could see the number of families have been increased from 232, which is in taxation system of classification, to 416 families. And this increase in the number of families is based on the basic principles having monophyletic condition. Major features in this system of classification is the disappearance of the certain division which we consider in the previous system of classification, like monocot and dicot. So the dicot has been completely aborted, whereas the monocots have been placed in between the basal angiosperms and the eudicots. So basically, some of the ranks which is being followed in the previous system of classification, like classes, divisions, have been abolished, and a new concept called clades have been introduced. Example, the magnolids. Besides, two major clades have been introduced. These super rosids and super astids. The relative contribution of different clades includes eudicots shares about 75% of the angiosperm, whereas the monocots comprises of 23% of angiosperm, and magnolids 2%, and there are around 250 families. They contribute about different clades. They are mostly they are the paraphyletic. Some interesting points in the system or FPG system of classifications, where Asclepidaceae has been merged with Apocynaceae. And the families like Bombycaceae, Serpuliaceae, Teliaceae have been merged with Malvaceae. The family Chenopodiaceae have been merged with Amaranthinaceae. On the contrast, Brassicaceae was split into three different families: Brassicaceae, Caparaceae, and Cleomaceae. Euphorbiaceae is being split into another four families: Euphorbiaceae, Finlandaceae, Picrodendraceae, and Putrangiaceae. Isoaceae has been split into two different families: Monoginaceae and Hygescaceae. So these are all based on the phylogenetic system of classification. So the largest families in the APG system of classifications are Orchidaceae with 27,800 species, Astaceae 23,600 species, followed by Fabaceae, Rubiaceae, and Poaceae. So in this system of classification, also they followed certain basic principles with regard to floral parts. Normally, the evolution has taken place from simple to complex nature. So from floral parts that are many to few, and floral parts that are separated to fused, or that means from polypetalate to gametopetallate. In terms of ovary, the evolution is from superior to inferior ovary. In terms of the symmetry, the radial symmetry has evolved into bilateral symmetry. And in case of a pollen aperture, the monosulcate pollen nature has led to the tricorporate nature of the pollen grains. So in this case of this phylogenetic system of classification has used a lot of genetic markers like RFLP, RAPD, ISLR, etc. So the RFLP went for restriction, fragment, and the polymorphism. It is a non-PCR based method. It was introduced in 1980, and it is used. It has been the basic principle is the hybridization of a DNA with hybridization of this DNA with the labeled DNA. Which is used, which is been done with the help of southern plotting, and the DNA have been digested with restriction endonucleases. The percentage of hybridization is been used to the similar, taken it as a similarity. And another marker, RAPD, random amplified polymorphic DNA, which was introduced in 1990. these are used as a dominant marker because that requires no prior knowledge on the taxa that you are working at. And similar marker like inter-symbol sequence repeats, and these repeats are introduced in 1984. It is used to study the polymorphism in the symbol repeats that is found in the DNA. The simple repeats are otherwise called as the microsatellites. Another more important marker that has been used in the is amplified fragment length polymorphism. It was introduced in 1995. It's the advanced version of RFLP. Here, this also has been used to have a higher reproductive capacity and sensitive to detect the polymorphism. What is being present day used is another marker called internal transcript spacer (ITS). It is a spacer gene situated between the small and the larger subunit in the ribosomal RNA gene. It has got a high degree of variation between closely related species. And it is easily detected. It is easily detected even with a smaller quantity of DNA. So nowadays, scientists they use the ITS marker for system of classification. There is another set of markers that have been used based on the chloroplast. So chloroplast is the independent genome. It replicates. I mean, it regenerates separately other than the nuclear genome. So there are certain regions in the chloroplast that have been considered as some markers, like RBCL, mature K, tRNF L, and tRNF, etc. So RBCL, the ribulose 1,5-bisphosphate carboxylase oxygenase enzyme, it is being used in the carbon fixation, and this is the most abundant enzyme present in the angiosperm. So it consists of about 1,400 base pairs located in the chloroplast, and it codes for the enzyme ribisco, that is the ribisco. RBCL has got a high similarity, and it exhibits low level of a variation, which helps to differentiate the taxa of a higher rank, because there has a low variation and mutation. Mutation in terms of in this gene is very rare, very rare. And mature K, MAC-K, it consists of 1,500 to 1,700 base pairs. It is also a plastic gene. It codes for enzyme maturase, and this protein is used to splice the group 2 introns. It's a self-splicing enzyme. It was first described, first characterized in tobacco in 1985. And this domain is found in all three domains. This mature K can be noticed in the all three domains: archaea, bacteria, and eukaryotes. And it has a high rate of substitution, highly evolved compared to RBCL. Another chloroplast marker like tRNA L and F, it consists of 250 to 800 base pairs. It also locates in the chloroplast. To aid this system of classification, the scientists have used a number of phylogenetic softwares, and the number of softwares are available in the website, in the net. But I have given you some important frequently used softwares like Phylip, Pop, Mega, Relmel, Mr. Basi, and Tree Buzzers. So all these techniques, markers based on these techniques, has evolved to use applied in the phylogenetic system of classification. Now I will move on to few examples. The APG4 system is the one system where phylogeny is applied for the entire organisms, and here I am going to present you with a few examples where it is being used below family level. So the RPG system, they are insisting upon up to family levels and above family levels, whereas the slides that follows here, I'm going to give you some examples of phylogenetic application below family level. So the cucurbit members have been studied with RAPD markers. And based on these studies, we'll be able to identify the taxa cucurbit of maxima based on the RAPD markers, whereas other members that have been included in the study could not be identified because they showed a lower level of similarity. The legume phylogeny working group worked on the family Fabaceae using the molecular markers MAC-K, RBCL, and tRNA L and F, and they have divided the family Fabaceae into 6 subfamilies, beginning with the Cecilioidae with 12 orders and ending with the Papilionoidae with 503 genera. So they are able to differentiate into 6 subfamilies. Earlier it was with 3 subfamilies like Papilionaceae, Cisalpinoidae, and Mimosoidae. But based on the phylogenetic studies, they are able to divide into 6 subfamilies. The family Mimosoidae have been merged with Cisalpinoidae. So that all the genus included in the earlier Mimosoidae members have been merged with the subfamily Cisalpinoidae. So molecular application at the infrageneric level, the genus Bloom, it comprises of 30 species in China, and it was divided into 3 sections based on ITS and tRNA LFN sequences. The 3 subsections are Paniculatae, Semivestidae, and Macrophyllae. And this is the differentiate these 3 subgenus. The yellow color is Paniculatae, green color is Semivestidae, and the blue color is Macrophyllae. The genus Impatiens belonging to the Balsaminaceae is represented by 260 species in Madagascar. Of these 260 species, they have studied 33 species, and they have ended up in delimiting the genus Impatiens into 2 subgenus based on the 2 nuclear regions. So these 2 subgenus includes Impatiens and Trimorphopetalum. Of these 2 subgenus, Trimorphopetalum is a monophyletic, and the Impatiens still remain as a polyphyletic. And this is the phylogenetic tree showing the 2 subgenera Impatiens and Trichopolypetalum. Phylogenetic studies at generic level. Phylogenetic study have been done to delineate the Medicago, Trigonella, and the Melitotis all belong to the family Fabaceae based on the molecular data, and they have used about up to 21 species. Of the 21 species, 16 species they were able to delineate, and the remaining becomes polyphyletic. Based on the study, they recommend that Medicago falcata to be treated as a subspecies of a Medicago sativa. And this is a phylogenetic tree study done in the Medicago and Melilotus, Melilotus. The genus Carcorus belongs to the family Malvaceae, has been studied based on the ITS sequences in 42 different species. Based on the study, it recommends that the Carcorus must be merged. All the Carcorus species must be merged, and that has evolved from the Africa and spread to other parts of the world. Based on the Carcorus species has been evolved from the genus Gruvia. The genus Dasyphyllum has been studied from South America, and it is one of the diverse group of Astraceae comprising of 33 species. It has been traditionally divided into two subgenera, namely Orchidasyphyllum and Dasyphyllum. A phylogenetic study was performed based on the tRNA-L and F, PSPA and H and ITS region. Based on the study, they have evolved their Orchidasyphyllum elevating to a generic level. So the subgenus Orchidasyphyllum has been elevated to genus Orchidasyphyllum, where the Dasyphyllum remains as a different genera. There is a whole lot of confusion between the genus Syzygium and the Eugenia. So in order to evolve, in order to resolve one Eugenia species, Eugenia boerlagi from Amboinier, this genus, this species, Eugenia boerlagi, been transferred to Syzygium boerlagi by Bowers. So the scientists have been taking up a phylogenetic study whether this move from Eugenia to Syzygium boerlagi was is correct. So based on the phylogenetic study using ATAPB and RBCL intergenetic spaces, they have concluded that it is better that Eugenia boerlagi is placed or considered as a Syzygium boerlagi because there is a lot of similarity of Eugenia boerlagi with Syzygium than the genus Eugenia. Phylogenetic study at specific level, the genus Acacia is considered to be polyphyletic, and scientists have been recommending that it should be divided into at least 7 genera. The work has been done in the Acacia species of South Arabia, and they have divided into 3 genera. So instead of 7 genera, what are species available in South Arabia, they have divided into 3 genera: Acacia, Senegalia, and Weichelia. So based on this division of Acacia into 3 genera, the Acacia nilotica, which is common in India, has been changed into Weichelia nilotica. So the correct binomial of Acacia nilotica is now Weichelia nilotica. Phylogenetic studies also have been used in delimiting new species. So work has been done in the Habenaria sandigorensis from Nepal. They have introduced this new species of Habenaria from Orchidaceae based on the molecular studies in combination with the morphology. the application of molecular studies in the barcoding of life forms. So molecular data also used in the barcoding system. The barcoding is nothing but a special level of identification where DNA is being barcoded in the early of 2000. So 4 primary gene regions have been used for barcoding: RBCL, MAC-K, tRNAH, PSVA, and ITS. And these have been generally agreed markers or standard markers for barcoding of angiosperm, specifically angiosperm. So based on this, after introducing the barcoding of systems, a number of work has been done. So Mauro et al. 2019, they have done the barcoding of Dipterocarpus genus. They have used MAC-K, RBCL, and tRNAL and F regions for their study. Similarly, there is another species, Dimonius salcidoi, which has been barcoded in 2016 by Faustin. This species is a critically endangered tree from Micronesia island near Australia. A barcoding of tropical trees. The barcoding of tropical trees have been done by Hume in 2015 using markers like RBCL, MAC-K, besides ITS and tRNAH. So these are, I have given a lot of, there are a whole lot of work has been done. It is available, but I have picked up a few of them to show how a phylogenetic approach has been applied for the entire angiosperm, as well as a few at a lower rank, like at species level or family level, etc. So here I am going to present a few studies that we have undertaken. So when we do the phylogenetic studies of, the taxonomic studies of angiosperms in various parts of Tamil Nadu, I have come across that there are certain taxa have been synonymized. And these certain taxa are particularly they are endemic taxa, so that it has gained attention in me. So I have started to work with using or applying the phylogenetic studies, whether it could aid in delineate again or reinstate certain species. So for example, Capparis incanescens been merged with Capparis separia. The genus Capparis, the species Capparis incanescens is endemic to peninsular India, and whereas the Capparis separia are widely pantropic. So they have merged these endemic species Capparis incanescens into Capparis separia based on a revisional work done by Jacob in 1965, where he revised the Southeast Asian Capparis species. Similarly, the Bleipharis molliginifolia have been synonymized with Bleipharis integrifolia, and Cucumis callosus in the synonymized with Cucumis mello, and Melotheria umbilicalis synonymized with Solina umbilicalis. So whatever species that we see, like Bleipharis or Cucumis, they are mostly endemic species, which have been synonymized with more pantrophic species. So this has got my attention, and that I wanted to come up with some of my colleagues who wanted to work on with the synonymous taxa. So this is Capparis incanescens. You could see both the species, Capparis incanescens and Capparis separia. As I told you, one Capparis incanescens is endemic to peninsular India, whereas the Capparis separia is a pantrophic. So they have merged together. So we have done a phylogenetic study and we are able to separate them based on the phylogenetic study that Capparis incanescens is genetically different from the Capparis separia. So now the Capparis incanescens was introduced by DC. He has D. Condole. He has taken the specimens from peninsular India, and that endemic species now being reinstated into Capparis incanescens and separated from Capparis separia. Similarly, this is a Blepharis molliginifolia, and this is a Blepharis integrifolia. So again, the revision of South African flora, the scientist Wallensen, he has merged Blepharis molliginifolia with Blepharis integrifolia. This Blepharis molliginifolia, as per Bentham and Hooker and the floristic work of Flora of India, is restricted to India and the allied countries like in Southeast Asia. Whereas the Blepharis integrifolia is endemic, or not endemic, but it is found distributed in the African continent, mainly in South Africa. So when they revised the flora of South Africa, they have merged Blepharis molliginifolia with Blepharis integrifolia. Recently, I happened to collect specimens of Blepharis integrifolia. While trying to identify, I have seen that this Blepharis integrifolia has been merged with Blepharis molliginifolia, and I have worked on that using phylogenetic study, but I could not present the data because it is in the process of a publication. So I am very sorry, I am not presenting the data of a phylogenetic study here. So similarly, another species called the Cucumis mello and Cucumis callosus. The Cucumis callosus, or otherwise called as the Cucumis trigonus, which has got a distribution in India and some parts of other countries related to India. Whereas Cucumis mello is a cultivar species, but Cucumis mello again is found in wild also. So Cucumis callosus have been merged with the Cucumis mello. Now the Cucumis callosus is not it's a synonymized with the Cucumis mello. So this work is also under publication. So I'm sorry that I could not publish the phylogenetic tree based on this. So we are able to differentiate based on the phylogenetic tree that Cucumis callosus is distinct from the Cucumis mello based on the molecular data. So they are distinct. Genetically, they are distinct. In addition to that, we have also applied the phylogenetic study to delineate a few new species. So this is a species, Cydrax cadalurensis, recently published in Phytotaxa. We have applied the phylogenetic markers in addition to morphological character, and this is the phylogenetic tree of Syzygium species, which shows that the Syzygium cadalurensis is being distinct from the Cydrax dicoccus. So the Cydrax dicoccus is mostly found in the lower elevation of Eastern Ghats and Western Ghats, and it is different from the Cydrax umbellata. So Cydrax umbellata is found only in the higher altitude. So in India, there are about 5 species, in Tamil Nadu, there are 5 species of Cydrax. And we are able to collect Cydrax umbellata, Cydrax dicoccus, and one species is restricted to the hills of Tirunelveli, so we could not get it. But based on the whatever collections we could have, we have done the phylogenetic study. So that help to delineate or to help to delineate Cydrax cadalulensis from closely related species of Cydrax dicoccus and Cydrax umbellata. So we have used the MATK, RBCL, and the ITS regions of the DNA. So there are few more species are under publication, but so I am not sharing it here for the reason of it won't be an ethical reason. So once again, I like to thank the organizers for providing me an opportunity to share with you or to be with you the small work that you have done. So we have just began for past 3 or 4 years. We have been working on this application, phylogenetic application in solving few taxon and mostly to delineate new species and to delineate or to uh, reinstate few species that have been already synonymized. Thank you one and all, and also I thank you for patient listening. If you have any clarification, you are most welcome.
Thank you very much, Dr. Susiraj, for your wonderful presentation. There is a question from Mr. Natarajan.
Yes, sir.
Which system of classification of plants is more reliable now?
Okay, thank you. So I am not very sure about what you mean by reliable, but now scientists they rely upon a phylogenetic system of classification because that is more recent and so every system of classification they have a basic principles. Every system of classification they have a basic principles. Either they are differentiate the taxa based on morphological features or anatomical features, etc. But now the most of the taxonomies consider the phylogenetic system of classification because this is based on the theory of evolution. So it is better that we group the organisms or taxa based on their in terms of their evolution rather than placing it based on their morphological features because there are number of morphologically similar species have been put together in one family. Number of species that are morphologically similar, but evolutionary they are different. Genetically they are different. Okay, so this concept molecular data have helped us to put all the species not only morphologically similar but also genetically similar. So this concept of phylogenetic evolution or phylogenetic system of classification comes based on that we share taxon. The many of the taxa share some portion of the genome as a common. So from this common genome, we species has evolved. As a species have evolved at a different time period of evolution. So it is better that we go for a phylogenetic system of classification. But for the purpose of arrangement of the herbaria or use of herbaria, still most of the herbaria they follow the Bentham and the Hooker system of classification because the data, the herbarium sheets are huge in number. Again, shifting them into different clades will be is it's difficult. It's difficult. Have I answered, sir?
Yes, sir. Yes, sir. In fact, systematics is the only field that can embrace all modern science developments.
Yes,
so
sir.
we have to apply as much as techniques possible to arrive at conclusions, whether it is for repositioning of a taxa or to identify a new taxa itself.
Okay,
That way, your explanation was very wonderful. And can you suggest some of the basic tools for audience for beginning taxonomy work in their area?
Thank you. Sir, it involves a lot of, from point of experience, I'm saying it involves a lot of techniques. We need to be acquainted with biotechnology, where you have to extract the DNA, and you have to amplify the gene, and you have to go for sequencing. And once you get the sequences, you go for doing the phylogenetic studies, and even after constructing a phylogenetic tree, you have to interpret it. So it is better that you collaborate. Always you collaborate with the scientists who are working at it, and you could take up any family or any genera, because it's enormous work has to be done. So with the phylogenetic system of classification which they have derived is not based on the 1,90,000 species that they have identified. No, they have taken only a representative species. So there is enormous opportunities available on any species or any genera, any specific genera or tribe or family could be taken up for a phylogenetic studies. The only difficulty would be these two. One thing is a collection of a species, because it's very difficult to collect it. And another difficulty would be the techniques available. So in India, we have very poorly developed lab where the techniques are available at our disposal. So where the minimum, the colleges and the universities. So now the extraction, sequencing, all these facilities are available with the central universities or highly sophisticated labs, which is a constraint. It is a constraint. Otherwise, we could take up any genus or any family for phylogenetic study. Am I clear, sir?
Yes, sir. Now the feedback form for the second day is launched. I'm reposting it in the chat box. The participants can fill up the feedback form separately for second day. We request the participants to type in their questions if they have. Dr. Rashid has raised his hand, but is not responding in chat. Whether he wants to speak to the.
Yes, sir. You are most welcome.
Again, there is a small problem in the feedback form. Please give us a minute to rectify it, and then we will launch the correct form. We shall wait for the organizing committee to identify the correct link for the feedback form for the day 2. Till then, we can have questions from the audience. We have a few more minutes to left behind. I have sent the direct message to Dr. Manoharan about day 1 feedback. If any one of you have not filled feedback form for day 1, you can contact us through direct message in the chat box. Then we will be able to give you the day 1's feedback.
Now the feedback is ready, sir.
Yes, yes, yes. Dr. Susiraj, sir,
Yes, sir.
you have got another question. Can you explain how far we can use herbarium for molecular identification of plants? Maybe you
Thank
can explain about using herbarium specimens for molecular phylogenetic.
Okay, thank you, thank you. As per the sequencing technology involved, uh, fresh plant specimens have been requested because the herbarium sheets, the extraction of DNA from herbarium sheets are is very difficult. So we get a very few small amount of DNA from the herbarium specimens that too are highly distorted. So because we are going to use pesticide, we are using insecticide, and we are poisoning it with a lot of chemicals. So the material available in the herbarium be minimally being used, very minimally be used, but there are in few works are available from other countries, especially not from our country. Because whenever we submit samples for sequencing or DNA extraction, they always ask for fresh specimens because they get high amount of DNA which can be used for PCR and amplification. Otherwise, the DNA gets fragmented or distorted, which is going to hinder our results. So, if you want to get a better result, always we should go for a fresh specimen. Rare cases, DNA being extracted from the herbarium deposits, but that is not much useful. So far, it is not being much used because of the low amount of DNA being extracted and amplified.
Yes, sir. Is there any DNA friendly preservation methods while preparing herbarium we can adapt? Because the formalin can degrade the DNA.
Formaldehyde or spirit, methylated spirit, that's what normally they use it for preservation. Methylated spirit or formaldehyde, both of them are harmful to the DNA content. Maybe to some extent, you can preserve the material maybe 1 month or 2 months, 3 months in liquid nitrogen. Otherwise, I don't think we don't have at present we don't have any other alternative. So that is also another difficulty. You have to collect the specimen fresh when you go for a study. So some specimens, some taxa are not available at a specific point of a year. So that would give us a difficulty in collecting the specimen. So preservation as such, they do it only store it in liquid nitrogen. That too for only a few months, only for a few months.
There is a question from Adil Farooq.
Yes, sir.
How can we barcode a novel sequence? I think he's asking about how can we find out novel sequences for barcoding the plants? Like you have pointed out commonly used barcode sequences.
Yeah.
He wants to know novel sequences.
I am not.
Can we identify?
Yes, sir. I am not technically competent to speak about that markers because I am solely depend on the markers available in the market. So I am not competent enough to say which markers can be used other than what is available in the system. That needs to be worked upon. I'm sorry, I cannot be able to explain.
Okay. We shall look for newly available universal marker.
Yeah, that's why I'm saying.
Publications, yes, sir. We have to look for new publications that come up with new markers. Then we can adapt it.
Yes, that's what I too feel. You have to look for, or you have to work on developing new markers, for which I'm sure I am not a competent at present. Maybe later you can develop.
And there are so many messages greeting that your session was wonderful, Dr. Susira.
Thank you very much, sir. I am once again, I thank all of you for the opportunity that is being provided to me. Thank you very much.
Shall we wind up, sir? If there are no more questions for Dr. Susira, we winding up of the program. Sir, you can stop sharing your screen, sir. In that case,
Thank you, sir. Thank you.
thank you very much. Dear participants, delegates, respected resource persons, and everyone gathered in this cyberspace, I am very happy to sum up what we have learned for the past 2 days. This is going to be a short seminar report. For want of time, I will make it as short as possible. Yesterday, in day 1, session 1, we had Professor K. Praveen Karan. He spoke about phylogenetic diversification studies in endemic lizard, Hemidactylus geckos. It belongs to India. It has some interesting revelation about adaptive radiation in this animal. And he pointed out certain activities at college as well as school level in studying molecular evolution. For those who are interested in taking up this kind of studies, generation of his take-home message was, generation of data is quite easy, but interpretation is quite difficult. For this, he offered a chance to learn about molecular phylogeny by enrolling yourself for workshops conducted by Indian Institute of Science. So look for announcements about molecular phylogeny workshops. As pointed out by Professor K. Praveen Karan, he was giving quite extensive example about how basic statistics and phylogeny can give you out interpretation about evolution of animal system. In the second session, Dr. R. Ramasubbu, who is an assistant professor from Gandhigram Rural University, he pointed out important things about the biodiversity of Western Ghats. He gave extensive coverage of plants as well as animal diversity in Western Ghats, which is an important biodiversity hotspot in India. And he focused about forest types, vegetation types, formations, important formations of Western Ghats. And he pointed out rare plants. He focused about endemism, endemic plants of Western Ghats. He also gave some insight about rare, endangered, and threatened taxa. He gives some idea about how impatiens diversity can help focus on the ecological stability as well as studying the progression in ecosystem. And he gave a bonus awareness about poisonous snakes of Western Ghats, how they can control rodent and pests in agri-ecosystem adjoining to the forest ecosystem. He added note on king cobra as a flagship species, which can control the population of other venomous snake species. So out of his bonus lecture, like we were able to understand, the snakes are all not at all only bad animals, but they have also some positive sides, like the venomous snakes give important proteins in their venom that can be used in times of open heart surgery. And today we started the session with another resource person, eminent resource person from Northeastern region. Yesterday we had a very good lecture on Western Ghats, and today Dr. Lata Rangan pointed out about the molecular diversity in the region of Northeastern India, which is another biodiversity hotspot, important biodiversity hotspot in the world. She gave a wide coverage of plant group gingiberales, and she has carried out extensive studies ranging from cytokaryology to flow cytometry. The take-home message was gingiberales being vegetatively propagated plant group show high scope for carrying out evolutionary studies, and this is only possible if both rigorous field work and modern science are amalgamated in research methodology. Systematics is the only field of biology that can embrace any modern analytical tool that is developed in science. And now, just now we have finished listening to Dr. S. Susera, who is another eminent scholar in the field of taxonomy. He has given wide application of wide information about application of molecular data in angiosperm systematics. He started with very basic information ranging from what is taxonomy, what is systematics, and what are all the different systems of classification. And he gave very good idea about higher degree of applications which are applied for repositioning of taxa in the systematic position, as well as renaming of taxa and evolution of new groups of species. So we come to know about this molecular evolution and its application in taxonomy as well as ecology in these 2 days, fruitful days. I once again request all the participants to fill up their feedback forms for separately for day 1 as well as day 2. Give us your positive feedbacks. If at all you find anything to be conveyed to us, you can right away mail us or you can direct message to us before the meeting ends. If the meeting has ended, if you want to convey something to us, you can write email to us. So with this, I would like to propose vote of thanks for this seminar. I would like to thank God Almighty on behalf of the organizing committee members as well as the delegates, resource persons for giving us wonderful opportunity to participate in this 2-day long national webinar on molecular evolution, taxonomy and ecology. I would like to thank on behalf of the Department of Botany, I would like to thank the DBT Government of India for funding this seminar by way of DBT Star College Scheme funding. I would like to thank all the members of our management committee of Jamal Mohamed College, secretary, assistant secretary, treasurer, and director, honorary director, and the directors of hostel administration, and all other members of the management committee for their moral support and constant source of encouragement in organizing this kind of programs in the college. And I would like to thank our beloved principal for his encouragement in motivating as well as being part of all our endeavors in this department. And I would like to thank vice principal, additional vice principal, dean of science, dean of research and consultancy, and all other faculty members from botany department as well as the sister departments for actively participating in this workshop. I would like to thank all the delegates, each one of you, for attending this event and making it as a grand success by logging in time as well as giving us your positive feedback in way of active participation. And also, I would like to inform all the participants that the certificate will be issued to you by way of email. It will take a week's time. If you have not received after a week, you can directly inform us by way of email, as well as we can contact the organizing committee members for receiving the certificate. If you have not received the certificate in the inbox, kindly check in with the spam box, because the certificates are sent through bulk mail. It has a high possibility of landing up in spam folder instead of in inbox folder. So with this, we would like to conclude this session. I once again remind those who have logged in lately to fill up the day 1 and the day 2 feedback forms separately. I would like to thank all the authorities of DBT Government of India by way of DBT Star College Scheme funding to Jamal Mohamed College, as well as all the departmental coordinators of Jamal Mohamed College for being instrumental in organizing this 2-day national webinar on molecular evolution, taxonomy and ecology. Thank you very much. If you have any queries, you can directly message to us, and you can email to us also if you have any clarification. The certificates will take a week's time. After that, if you have not received the certificates, you can directly contact us. Please check the spam box once again. Instead of looking into inbox, you can check always into the spam box, because the email will be, the certificates will be sent by bulk email. So it has a high possibility to land in spam box. Some of you have asked about the certificates in the direct message. So this is what I wanted to convey to all of you. I express my sincere thanks to each and every one of you who have sent their message to all the organizing committee members as well as the speakers. Thank you very much for all your encouraging words. Thank you very much.